Dr. Alper's contributions to our division and to the subspecialty of gastroenterology. So our tradition is that faculty gather to nominate a prominent physician scientist, and this year's unanimous selection was Anna-May Diehl from Duke. So a little bit about Professor Anna-May Diehl. So she attended Georgetown undergraduate and medical school. She trained as an internal medicine resident and a fellow at Johns Hopkins, and then joined the faculty there at Hopkins, rose through the ranks to professor of medicine, and was recruited to lead the GI division at Duke, where she is currently the Florence McAllister Professor of Medicine. She's a physician scientist with extensive lab-based research activities that focus on basic mechanisms of liver repair and clinical translational research interests in both alcoholic and non-alcoholic fatty liver disease. Her basic research program has been supported continuously by the NIH since 1990. She's the principal investigator for the NASH CRN U01 clinical research program at Duke since its inception over 15 years ago. And she's also the site PI for the liver cirrhosis U01 network that began earlier this year. She has been elected to membership in the American Society for Clinical Investigation. She's received numerous awards, including an NIH NIAAA Merit Award, the Duke Distinguished Faculty Award, and the American Association for the Study of Liver Disease Distinguished Scientific Achievement Award. Among the many true accomplishments of Professor Diehl's distinguished career has been her commitment to mentorship. So she's trained over 70 individuals in her research lab and as chief of GI at Duke. She has served on multiple scientific review panels. She served on NIH, VA, AGA, and AASLD Career Development Award panels. And she will be the co-chair of the AGA AASLD Academic Skills Workshop in 2022. Dr. Diehl has been an associate editor or an editorial board member for all the leading journals in GI and liver disease, including Hepatology, Gastroenterology, Gut, and the American Journal of Physiology. She's received numerous awards and is currently a member of the Executive Governing Council for the International Association for the Studies of Cells of the Hepatic Sinusoid, as well as co-chair of the NAFLD Special Interest Group for the AASLD. So it's an honor and a privilege to introduce my friend and colleague, Anna-May Diehl, as the 23rd Alpers professor. She's going to give a talk entitled Fixing Fatty Livers. Anna-May, thank you so much for your patience and bearing with us, and we're looking forward to hearing your talk.
Nick, thank you very much. Can everybody hear me?
I can hear you fine, thank you.
Perfect, perfect. Well, we've overcome the greatest obstacle of the COVID pandemic for me is mastering the utilization of any kind of a Zoom link, but I think we've done it. So I am truly humbled to be invited to give this lectureship in honor of Dr. Alper. He is one of the giants in the field of gastroenterology. And so I will try to do him justice today by sharing with you some of the latest stuff that's coming out of my lab, but I hope that I can make this relevant to those of you who are not bench researchers and actually work in the clinic, as I do as well, trying to fix fatty livers. Next slide, please. I think, yeah, there you go. So I don't think I have to tell anybody that the world today is getting busier, less active, and fatter. And in fact, the statistics bear that out. Between 1988 and 2010, US adults reported no leisure time, increased in men from 11 to 44%, and in women from 19 to 52%. In fact, the CDC today says that only 41% of US adults are totally inactive, meaning that we do less than 10 minutes of physical activity a day. In parallel with that increase in sedentary lifestyle is an obesity epidemic. Since 1980, obesity prevalence has doubled in 73 countries around the world, and we now have at least 600 million obese adults and about 110 million obese kids. And paralleling that obesity epidemic is an epidemic of the metabolic syndrome. Now, the metabolic syndrome is really just a constellation of biology, really, that parallels visceral obesity. It includes hypertension, insulin resistance, dyslipidemia. Next slide, please. Population aging is exacerbating the public health impact of the metabolic syndrome. As shown in this JAMA paper from 2015, the overall prevalence of metabolic syndrome in the American population is now 33%. And in both males and females and all ethnic subgroups, the prevalence of the metabolic syndrome increases with age. And for people my age, it's pretty daunting because we know now that over half of women over the age of 60 and Hispanics over the age of 60 have the metabolic syndrome. Next slide. So why care about the metabolic syndrome? Well, it's thought to reflect a chronic inflammatory state with dysregulated energy homeostasis. And it's a major cause of death now because it is associated with tissue degeneration, eventual organ failure, and cancer. And I think people are well aware of that when it comes to heart disease and kidney disease, but only recently has it become evident to everybody that there's a liver disease that goes along with the metabolic syndrome, and that is non-alcoholic fatty liver disease, or NAFLD. NAFLD is a spectrum of fatty liver damage that strongly associates with the metabolic syndrome. And just like other tissue damage in the metabolic syndrome, it's characterized by progressive tissue degeneration that ultimately leads to organ failure and sometimes malignant transformation. NAFLD has become a major cause of cirrhosis and liver cancer in the United States and many other industrialized countries, but even poorer nations are beginning to experience this epidemic. And as such, it's become a leading indication for liver transplantation. So this is a bad disease, and we wish we could fix it, but the mechanisms that determine the susceptibility to the metabolic syndrome, NAFLD, and their sequelae remain poorly understood. Next slide. And so it's hard to fix something if you don't know where it's broken. And this is evidenced by the fact that many of the phase 2 and 3 randomized clinical trials that are testing novel treatments for NASH have actually ended up in the graveyard. And we still, in spite of a lot of effort, don't have any leading candidates. And so obviously we need to do more discovery work. And I'm going to share with you today some of the work that has been going on in my lab. I think we've made a discovery and that it might have therapeutic implications. And I'm going to talk a little bit about that. And then show you that maybe this isn't just a pipe dream. There might actually be some evidence for the efficacy of these putative therapies. Next slide. So let me start with sharing with you the discovery work. For those of you that are clinicians, please don't be daunted. There's not going to be too much mouse data, but I do need to show a little bit to explain how we got to where we are. Next slide. So my lab has been studying this developmental morphogenic signaling pathway called Hedgehog, and Dr. Davidson's group actually also did some work in Hedgehog. A hedgehog is active in development and many cancers. It was actually thought to be silenced in adult tissue because its major role is orchestrating tissue construction. And so obviously it has a role in organogenesis during development, and it becomes dysregulated in cancer. And therefore, the things that it regulates go awry. And one of the things Hedgehog does is it controls tissue construction by controlling proliferation, differentiation, and senescence in cells. It turns out that Hedgehog may be relevant to the metabolic syndrome because this pathway is exquisitely sensitive to metabolic control. And that's because the ligands and the receptors are directly regulated by fatty acids, cholesterol, and their derivatives. Deleting smoothen, which I've highlighted in the upper right in bold font, smoothen is a G protein coupled receptor that's membrane associated. And it is the signaling competent receptor. If you disrupt smoothened, you can block both canonical and non-canonical hedgehog signaling. So this gives us a tool or a way to manipulate this pathway. Next slide. And so we were playing around with this pathway, and we were surprised to demonstrate that disrupting hedgehog signaling in adult hepatocytes caused NAFLD and the metabolic syndrome in chow-fed mice within a week. Fat in the liver went up fourfold. And along with that, we saw an increase in serum triglycerides, hyperinsulinemia, and insulin resistance. So just knocking out that one protein in hepatocytes was sufficient to cause fatty liver and the metabolic syndrome very rapidly. Now, remember, I told you that the metabolic syndrome is associated with tissue degeneration and that NAFLD progresses as the liver degenerates. And so we looked at how this impacted the liver's ability to regenerate. And what we found was that this knocking down of smoothen diminished hepatic regenerative capacity in mice as well. Next slide. So for the last little bit, we've been trying to delve into this a bit further. And I'll show you how we did this, but we were able to selectively delete this smoothen factor that regulates hedgehog signaling in hepatocytes. And that is going to allow us to say what happens when we do that in hepatocytes. And are these effects biologically plausible as triggers for the progressive degeneration that occurs in NAFLD? And if it's true in mice, is it also true in people? In other words, do hepatocytes with diminished hedgehog activity actually accumulate in human livers as NAFLD progresses? So I'm going to tell you about that for the next little bit of the talk. Next slide. So the way we did this in mice is we have these animals that we call smooth flox mice, and this means that the smoothened allele is flanked by sites that allow us to clip it out. So we can selectively clip out smoothened in whatever cell we want to if we have an appropriate way to target the deletion. And we can do that by injecting the animals with viral vectors that bear an enzyme called Cre recombinase that snips out alleles that are flanked by those sites. And then we can compare what happens when we take smoothened out compared to when we leave it in. And in order to do that, we treat animals with a control vector. So we did that. We waited 7 days, and then we isolated the hepatocytes. And then we took advantage of a novel technique that's really quite remarkable. It's called single cell RNA sequencing, and there are different platforms that you use. We happen to use 10x Chromium. And this allows us to get the transcriptome of individual hepatocytes. So you can imagine there are many thousands of hepatocytes in an individual liver. So you can get 1,000 replicates of your experiment, if you will. So it gives you very good statistical power to demonstrate small differences. So we did this to try to figure out what would be the effect of taking smoothened out in hepatocytes of a healthy animal. Next slide. And first, we wanted to make sure we had pure preps of hepatocytes. So this technique, you can ask it to look for markers of hepatocytes or markers of non-parenchymal cells. And we did have a few non-parenchymal cells in our prep, so we could eliminate further consideration using kind of in silico approaches. And that left us with 5 subpopulation of hepatocytes that you can see on the bottom left. And the most of the these hepatocytes were very pure. They were expressing hepatocyte markers, but you can see they didn't have any non-parenchymal cell markers. So we can focus our analysis on those cells, and that'll tell us exactly what happens in the hepatocytes, and we won't worry about its effects in any of the other liver cell types. Next slide. So as you can see, there were 5 clusters of hepatocytes shown in blue. And several of those clusters that were kind of small, clusters 2, 5, 3, and 4, it didn't really matter much to the transcriptome if we deleted smoothen or not. The blue and the pink bars of the knockouts and the controls are overlapping. But the largest clump of hepatocytes, which we call HEP1, did seem to be affected when we knocked out smoothen, and it segregated into a group of cells that had a transcriptome in the knockout that was different then the transcriptome and the hepatocytes from the controls. Next slide. So people in liver now are very interested about zonality, because if you look at the liver under the microscope, it looks like a big bland collection of hepatocytes, but actually they are quite diverse functionally. And we know that their functions distribute along the pattern of blood flow into the liver, which enters in the periportal area and then trickles through the sinusoids and leaves the liver around zone, what they call the pericentral zone of the liver. And other investigators had already used single cell approaches to define the gene expression profiles of cells in different parts of the liver lobule. So we layered that data over ours and learned that our HEP1 population, which was the big population of smooth and sensitive hepatocytes, really most resembled the hepatocytes in zone 2, or the zone between the periportal hepatocytes and the pericentral hepatocytes. It's interesting that zone 2 hepatocytes now seem to be the most developmentally, if you will, flexible. They seem to be able to reprogram themselves to look like either zone 3 hepatocytes if they need to, or zone 1 hepatocytes. So this was quite interesting. Smoothen was most active in these mid-zonal hepatocytes. Next slide. So then we drilled down a little bit further, and we wanted to say, well, could we further characterize that subpopulation of cells? And here we got really lucky, because it turns out that there was one clump, which we called C1, which had low abundance of smoothened alleles because it was almost entirely comprised of hepatocytes from the control animals. On the other end, C3, shown in blue, was 100% composed of cells from the controlled animals. So we had some cells that had low smoothing because they were all from the knockout, and some cells that had high smoothing because they were all from the controls. And so you would expect that the cluster that had high smoothing would have the highest hedgehog pathway activity. And in fact, when we used unbiased approaches to look at that by looking at gene signatures, we found that actually the highest hedgehog signaling was associated with cluster 3, which was made up of 100% from cells from the control animals. Next slide. So then we could play around a little bit more and develop a gene signature that identified these cells that had high smoothened expression and therefore high hedgehog pathway activity. And we reapplied it to the data set, and it effectively discriminated the cells that had high smoothened, high hedgehog from the cells that had low smoothened, low hedgehog. So all this work that I just described to you was really done in preparation for trying to ask, could we then apply what we learned in these mouse models to understand what was happening in patients? Next slide. And so I'm a clinician. We do a lot of liver biopsies. We see a lot of NAFLD patients at Duke, and we've created a biorepository. And so we took 378 subjects from our Duke NAFLD biorepository that we had liver biopsies on. All of these biopsies had been scored for NAFLD activity and fibrosis stage. And we did a bulk RNA sequencing on their livers to identify the transcriptomes of the whole liver. And these patients that we evaluated, we could cluster depending on the severity of their liver disease. So we had 69 people that, even though we thought they had NAFLD, actually on the biopsy, they had only non-specific changes. And so they were our controls. The other 299 people had NAFLD, and we could segregate that NAFLD cohort into 3 subgroups based on fibrosis severity, about 100 in each group. So about 100 with low or no fibrosis, some with intermediate fibrosis, and then a group with high fibrosis cirrhosis. We had bulk RNA-seq data from all of these people. And now we can apply that signature that we made in mice to the human bulk RNA-seq data and ask, what happens to the hepatocytes that have high smoothened or high hedgehog activity? We would expect, if what we see in humans is like mice, that we should get fewer smoothened high hepatocytes as the disease progresses, and more hedgehog depleted hepatocytes as the disease progresses. So what do we get? Next slide. Well, we got what we thought we would get. The smoothened high signature was depleted in human NAFLD. And so if you look at this slide, what you can see that compared to the controls, which had the highest smoothened high signature, all of the NAFLD patients shown by the 3 boxes on the right had lower amounts of smoothened high cells, and the numbers of smoothened high cells decreased progressively as fibrosis stage increased. Next slide. And so what that means is that since the hepatocytes with high smoothened activity were going down, the hepatocytes with low hedgehog activity were accumulating. So it looks like humans and mice are very similar in this regard. When we knock out smoothened and deplete high hedgehog hepatocytes in mice, we get NAFLD in the metabolic syndrome. And in humans who have NAFLD in the metabolic syndrome, they have low numbers of high hedgehog cells and high numbers of low hedgehog cells. Next slide. Okay, so it happens. Does it make a difference? What is the pathobiological relevance of having your liver become populated by cells that have low hedgehog activity? Next slide. So to answer that, we started to characterize the low hedgehog hepatocytes. And we were able to do this because we were using this clever single cell RNA sequencing tool, and we could apply various algorithms that tell us what processes and pathways are differentially regulated. And lo and behold, what we found is that compared to the high smoothened hepatocytes, the smoothened low hepatocytes demonstrated repression of processes that are involved in fat metabolism, insulin signaling, and mitochondrial function. We drilled down into this a little bit further and were surprised to discover that one of the major things that differentiated hepatocytes that have high smoothened activity from hepatocytes with low smoothened activity was actually expression of mitochondrially encoded electron transport genes. And that makes sense because the electron transport chain is what regulates oxidative phosphorylation and ATP production. And we found that those pathways and processes were repressed in the smooth and low hepatocytes. To further confirm that, we actually isolated hepatocytes from wild type mice and used seahorse experiments where we treated the cells with control drug or a drug called cyclopamine that directly inhibits smoothen. And what we found is that in wild type cells, if we inhibit smoothen pharmacologically, we see a suppression of oxidative phosphorylation and ATP production. So all of the data then was consistent with the fact that these smooth and low hepatocytes have dysregulated fat metabolism, reduced insulin signaling, and mitochondrial dysfunction. Okay, that's true in mice. Does a similar thing happen in people? Next slide. Well, it does. And this is just one paper that nicely summarizes a bunch of data from multiple labs showing that mitochondrial dysfunction is linked to reduced insulin signaling and dysregulated fat metabolism in people. So mitochondrial dysfunction is an interesting thing. It obviously is going to impact metabolism, but we know that mitochondrial dysfunction also causes oxidant stress. Next slide. And so what we wanted to see is, do these hepatocytes that have mitochondrial dysfunction, the smooth and low hepatocytes, do they have oxidative stress? And in fact, the analysis suggests that they do. They respond to oxidative stress, hydrogen peroxide catabolism, apoptotic signaling, and DNA damage were all significantly upregulated in the smooth and low hepatocytes. We confirmed that by looking at the livers of animals that had smooth and depleted in hepatocytes or not, and we stained them for a marker of DNA damage that detects double-stranded DNA breaks. And you can see that the nuclei are pink in the smooth and low hepatocytes, meaning that those hepatocytes had accumulated DNA damage, and we didn't find that in the wild-type cells. In other smooth and knockout and wild-type animals, we actually isolated the hepatocyte DNA and looked at the length of the telomeres, and telomeres shorten when you have DNA damage. And what we find is that the telomere length is significantly reduced in the smooth and depleted animals. Next slide. This too seems to be similar to what occurs in human NAFLD. It's known that early in the disease, when you have just fat in the liver, but not too much injury or inflammation or liver cell death, that the mitochondria have are functional, and they seem to have adapted to the energy excess that they're being asked to cope with. But as the disease progresses, these mitochondrial adaptations are lost, and this is associated with a progressive increase in reactive oxygen species production, which you can see in the graph on the upper right, and the increase in ROS production in human NASH is associated with an increase in oxidative DNA damage. So again, mice and humans are looking very similar. Next slide. What's this all mean? Well, tissue degeneration occurs in aging. And I told you in the very beginning that aging is associated with an increase in the metabolic syndrome. Could this all mean that when you delete smoothen, you're accelerating aging? So to evaluate that more directly, we were fortunate to come upon a paper that had just been published by Zhang et al. In 2021, where they had actually used single-cell RNA-seq analysis to profile the transcriptomes across the spectrum of life in mice from just about every tissue in the animal's body, including the liver. And they developed a murine aging signature. And it turns out that the murine aging signature, when they compared the gene expression in 30-month-old mice to 3-month-old mice, significantly overlapped with that of our smoothened, depleted mice. And in fact, when we looked at the entire gene expression signature, we found a very high correlation. The R value was 0.66, highly statistically significant. So it looks like the smoothened low hepatocytes are phenocopying the transcriptome of old hepatocytes. Next slide. And I told you before that the metabolic syndrome and the liver disease associated with the metabolic syndrome, NAFLD, increase with age. Next slide. So in summary, what I've shown you from the preclinical study so far is that we've learned that pathways that orchestrate organogenesis and carcinogenesis that were once thought to be turned off in healthy tissues actually remain active, and that sizable populations of hepatocytes in healthy adult mouse and human livers have this hedgehog pathway. Hedgehog signaling assures hepatocyte insulin sensitivity and maintains lipid homeostasis and mitochondrial function. Furthermore, loss of hedgehog inhibits hepatocyte insulin signaling, causes systemic insulin resistance, dysregulates lipid metabolism, disrupts mitochondrial function, increases oxidative stress, and promotes oxidative DNA damage. So it looks like inhibiting hedgehog signaling in hepatocytes significantly and rapidly accelerates cell aging, which we know is a major risk factor for NAFLD and the metabolic syndrome. Next slide. So now we have a new working model. And we know that healthy adult livers have a lot of metabolic flexibility and robust bioenergetics. And we would suggest that the evidence now is looking like something happens to decrease hepatocyte hedgehog activity early on in the metabolic syndrome. And it leads to the metabolic compromise of the hepatocytes, evidenced by insulin resistance, mitochondrial dysfunction, and oxidative stress. Over time, the liver becomes repopulated by bioenergetically depleted cells that have oxidative damage. And as you would expect, that would decrease cell survival. And the cells that do survive have had to do pro-survival adaptations. Some of those pro-survival adaptations resemble what we see in senescent hepatocytes. And we know that senescent hepatocytes have a very active pro-inflammatory and pro-fibrogenic secretome, and that is associated with the development of organ fibrosis and organ failure, and that the cells that can survive in that environment are often malignantly transformed, and therefore you would get the outgrowth of hepatocytes that escape this SMOO inhibition, i.e. liver cancers. Next slide. So if this is true now, this model would say that NAFLD and the metabolic syndrome are actual consequences of low hedgehog activity. Next slide. Now, I don't know how many of you have been following this story in the NAFLD field, but we've actually been saying the opposite thing for a lot of years, because we and others have shown that dying cells that are not yet dead make a lot of hedgehog ligands. And you can see in the upper right a photomicrograph of a human liver that was stained for sonic hedgehog. And sonic hedgehog is densely produced by ballooned hepatocytes, which are a hallmark of NAFLD. And in fact, we and many other groups have now shown that these hepatocyte-derived hedgehog ligands drive inflammation and fibrosis progression in NAFLD. So here we're saying high hedgehog is bad, but now I just told you low hedgehog is bad. So how do we reconcile that? Well, I think what I just showed you is that hepatocyte SMU activity and hedgehog activity are low in NAFLD, but the other liver cells remain hedgehog responsive. So these dying but not yet dead hedgehog impaired hepatocytes are actually making hedgehog ligands. And that is being perceived by the other types of liver cells that are involved in wound healing. And you get an expansion of that hedgehog sensitive wound healing population, which includes progenitors, immune cells, capillarized sinusoidal endothelial cells, myofibroblastic stellate cells. And they're frantically trying to send signals to rescue these hedgehog resistant dying hepatocytes, but it's too late. And there's a progressive loss of hepatocytes. And the more the hepatocytes are dying and lost, the more hedgehog comes out, the more these other cells are whipped into a frenzy. It's perpetuating futile cycles of repair that we recognize as chronic inflammation, fibrosis, progression, NASH, cirrhosis, and eventually liver failure. Next slide. So to state it a more specific way, actually NAFLD and the metabolic syndrome are states of hedgehog resistance in hepatocytes. Well, once you kind of get it down that crisply, it starts to help you focus on what you might do to fix this problem. Next slide. And these are the therapeutic implications. Next slide. So it's obvious, maybe NAFLD and the metabolic syndrome are kind of like type 2 diabetes, right? You have too much of a good hormone, insulin, but it's not working. And so what do we do for type 2 diabetes? We try to improve sensitivity to insulin. So maybe we should try to improve sensitivity to hedgehog. Sounds easy, right? But actually, I was a little bit deceptive in the beginning when I showed you that cartoon of the hedgehog pathway. I really oversimplified it. There's a lot of stuff that regulates the activity of the hedgehog pathway. And this is good in one sense. It gives us lots of things that we can look at and go after and see if we modulate them, whether we could fix hedgehog signaling. But I would say it's going to be complicated and a lot more discovery work is needed. Next slide. So maybe we should go for more low-hanging fruit. I mean, what we do know, after all, is that NAFLD and the metabolic syndrome are systemic diseases, and our data would suggest that they're caused by a loss of hedgehog signaling in hepatocytes. So how could a loss of hedgehog signaling in hepatocytes cause this whole messed up systemic energy homeostasis? Well, I think we have to remember that the hepatocyte is a master regulator of metabolism, and it does that by factors that it produces called hepatokines. And hepatokines regulate resiliency, i.e. metabolism, in many other tissues. So one could propose that maybe NAFLD, which has messed up hepatocytes, has altered hepatokine production, and that would lead to metabolic dysfunction and degeneration in tissues outside of the liver. They then would do things in a confusing way for the liver and might actually be fanning more liver cell injury, and you would get progressive NAFLD and a vicious cycle. So should we target the hepatokines? Next slide. In other words, could we fix the hepatokines? Would that fix fatty livers? What I'm going to show you now is just one example that sort of provides proof of that concept. By no means is this the only thing, but we do have, and other people do have interesting data about this one particular hepatokine called IGF-1 or insulin-like growth factor 1. IGF-1 is a hormone, insulin-like growth factor, whose synthesis is stimulated by growth hormone. IGF-1 promotes tissue viability, function, and growth, and it actually auto-regulates its own production normally, because when you make IGF-1, it feeds back and suppresses further growth hormone production, and therefore, when you have low growth hormone, you would reduce IGF-1 expression. So normally this thing functions fine, and there is a lot of circulating IGF, and in fact, 75% of the circulating IGF-1 in adults is produced by hepatocytes. IGF-1 receptors are more abundant in extrahepatic tissues than in the liver itself. And so circulating IGF, which mostly comes from the liver, is binding to IGF-1 receptors in a bunch of different metabolically active tissues, including skeletal and cardiac muscle, fat, pancreas, brain. And it apparently is very, very important in regulating IGF signaling, but also insulin signaling, because IGF and insulin bind to the same receptors. Insulin can bind to IGF-1 and IGF-1 can bind to insulin receptors, et cetera. So there's a bunch of binding proteins, 7 of them, that modulate the local bioavailability of IGF-1 as well as insulin access to IGF-1 receptors. And I'm going to tell you a little bit about IGF-BP7 because there's some good data now in humans saying that that factor it actually inhibits IGF-1 signaling and insulin signaling, and it promotes inflammation, and it does so in part by inhibiting insulin signaling. Importantly, other groups have shown that if you take IGF or you inhibit IGF signaling, that you can get degenerative diseases, and that if you correct that by supplementing IGF or restoring IGF signaling, you can reverse the pathology. So this is an appealing hepatokine because it actually has been shown to have functionality. Next slide. Well, so we looked at this in our smoothened low mouse hepatocytes, and what the pathway analysis showed, I've tried to highlight by the black arrow. You'll see that one of the most down-regulated pathways in the smoothened low hepatocytes, which are the hepatocytes that accumulate as fatty liver disease progresses, show a downregulation of IGF signaling. This is shown another way in the upper right, where we look at the smoothened low hepatocytes. And if you look to the left-hand side of the graph, what you'll see is the things that are significantly repressed in the smoothened low hepatocytes, and you'll see there's low significant repression of IGF-1 and receptor, and specifically low expression of IGF itself. If you grind down even further and do the single cell RNA-seq analysis, you'll see that the smoothened low hepatocytes are depleted of IGF-1 and enriched for IGF-BP7. Next slide. Well, turns out this might be important, because a group from Boston has published a couple of really interesting papers in the last several years. If you look on the right, they realized that HIV patients often have sarcopenia and frailty and NAFLD, and that these patients have low growth hormone and low IGF-1. So they did a clinical trial where they treated them with a growth hormone releasing hormone called tesmorelin, and they found that increased IGF-1 levels. And the increase in IGF-1 was associated with a loss of fat in the liver, so improvement in NAFLD, non-alcoholic fatty liver, an improvement in serum ALT, so presumably improvement in liver injury, improvement in NASH. And they found that there was less progression of fibrosis. So here in humans with progressive liver disease due to fatty liver, we find low IGF-1, and restoring IGF-1 improves NAFLD, NASH, and liver fibrosis. The group then went back and just published a paper a little while ago in Journal of Clinical Endocrinology and Metabolism, where they had done RNA-seq on the livers of the patients in this trial, and they found that the liver IGF-1 mRNA expression was low in the NAFLD patients, and it got lower as NAFLD severity worsened, very similar to what we found. Smoothened low hepatocytes accumulate, smoothened high hepatocytes, which are the IGF-1 producers, go down as NAFLD progresses. And they found that IGF-BP7 mRNA expression goes up as NASH progresses. And interestingly, that IGF-BP7, which suppresses IGF-1 activity, suppresses insulin sensitivity, promotes inflammation. Most of what circulates in the blood seems to come from the liver, because they found good correlations between the serum IGF-BP7 circulating mRNA and protein levels and what they found in the liver. So they're suggesting it might even be a biomarker. Next slide. So there is now evidence, I guess, that some of the hepatokines, which we know are dysregulated in the liver when smoothened activity and hedgehog signaling is inhibited, seem to be very important in regulating systemic metabolic homeostasis and health. How are they doing that? What are they targeting? Is there really something that we're overlooking here? What's the common thread? Next slide. Well, I wonder if it's the mitochondria. Because what I showed you earlier is that we had a lot of mitochondrial dysfunction in this smooth and depleted animal. The mitochondrial dysfunction gives you ROS, it gives you short telomeres, it happens with aging. Is this really the hidden culprit behind the metabolic syndrome, defective mitochondria? Next slide. And if so, does it make sense? Well, it shows you that if we improve smoothing, which we know helps mitochondria, we improve the metabolic syndrome. If we give IGF-1, which is depleted when smoothing goes down, IGF-1 improves mitochondria, it improves the metabolic syndrome. And in fact, some of the stuff we're already telling our patients to do, drink more coffee, exercise, take vitamin E, eat a healthy diet, also improves mitochondria. So maybe that's where we need to place our attention. Next slide. I'd like to stop here and thank all the people who did the work that I shared with you today. I hope I've gotten you thinking about NAFLD in a little bit of a different way. And I'd be happy to take more questions. And thank you, and thanks to my funding organizations.
Fantastic. Thank you so much, Dr. Diehl. And so sorry for all the trouble this morning, but we appreciate your resilience and for sticking with us here. We do have just a couple moments. I know in interest of your time, you're going to try to talk to our trainees. Let me pose a couple questions from the chat to you. One question here is, what is the actual effect size of, quote, signature enrichment in NAFLD patients versus control? They mentioned, I thought expression data was normally plotted as log fold differences.
Yeah, that's a good question. I think this whole issue of trying to use single cell signatures to deconvolute what we see in human disease is pretty much in its infancy in liver disease. As far as I know, there's only been a couple of papers that have done this so far. So I can't really answer that question. What we do know is if we were to look at the whole bulk RNA-seq data, we do see a change in the hedgehog signature that goes across the population. What we'll need to do to address your question more specifically will be to develop markers of the hedgehog low versus hedgehog high population, and then do immunostaining and other techniques that can now be used to actually track individual cells according to that gene signature, and then do some sort of quantitation of the human livers to see exactly what percentage of the cells are depleted. So right now, all we can say is that the pathway looks like it is turning off in hepatocytes, that the numbers of hepatocytes that have the pathway on seem to be reduced, and that this has functional significance as we walk back and forth between the data in the mice and the humans. But it's a good point.
Dr. Lister Millman compliments you, says, excellent lecture. Would you mind sharing the differences in the smooth and depleted hepatocytes in those patients with NAFLD, that is only fatty liver, versus those with NASH, the inflammation plus the fibrosis? When does a reversible condition become irreversible?
Yeah, thanks, Mauricio. Mauricio and I are old friends. We've known each other since we were fellows, and he was in Washington. I think that's a good question, and what the paper that I cited that had the human data in it indicates that early on in fatty liver disease, the mitochondria are experiencing metabolic challenge, and that they adapt to effectively handle that challenge. And apparently, most people, at least most young people, can remain in that state for many, many years. But there are factors that then conspire against the resiliency of the hepatocyte mitochondria, and they began to lose that pro-survival adaptation. And you start to get cells that have defective mitochondria, and defective mitochondria means more ROS, and more ROS means more damage, and more damage means eventually the death of the cell. And that seems to happen in the patients who have NASH. Can we rescue this? I guess that's the million-dollar question. But the answer is probably not all the time. And it'll be very important for us to try to learn, now that we know what we should be looking at, can we actually map and identify a point of no return? One would imagine if your liver is repopulated by cells that have totally dysfunctional mitochondria, that at that point, things that might work early in the disease would no longer work late in the disease. So I think this is just allowing us to get a little bit crisper target, maybe, as we try to understand the heterogeneity of the NAFLD population. Everyone has realized that it's heterogeneous. We just don't know why it's heterogeneous. Perhaps this explains some of the heterogeneity. And as we identify better markers for the cells that are undergoing these mitochondrial adaptations, we may be able to figure out which people would be amenable to which therapy. But I agree with you, Mauricio. I don't think that it's going to be, we can rescue everybody's mitochondria. Maybe there is a point of no return.
Right. Well, with that, in the interest of time, I think we'll call it, even though we have a couple more questions. Again, Dr. Diehl, just want to say thank you very much for joining us today. Thank you for honoring the David Albers lecture series. Dr. Davidson, if you're still on, do you have any other last comments?
Yeah, I'm still on. Anime, that was a great talk. Very provocative findings. And I think there's going to be a lot of questions, I think, in the group afterwards. So hope to catch up with you in a little bit of time on our separate Zoom call. Thanks again for a great talk.
Thanks, Nick. I want to pick your brain. I didn't get to share with you. There's a big changes in lipoproteins here.
Yeah, I think
you know how
maybe bile acid metabolism
or other are changing. APOs are going.
There you go.
Okay. Talk to you later. Bye.
Well, thank you again, Dr. Diehl. Take care.
Bye-bye.